Hello and welcome to WIP's Biochemistry. In this module, we will be discussing fatty acid metabolism, the part second, which is fatty acid biosynthesis. Now, why we need fatty acid biosynthesis? Because fatty acids are required as in different functions. For example, they form building blocks like phospholipids and glycolipids. They are part of fuel molecules. Being part of lipids. Then they are components of important molecules like glycolipids, lipoproteins, and precursor of second messengers. And fatty acids breakdown gives energy also. So this is very important. Fatty acid breakdown gives more energy than breakdown of a single molecule of glucose. Now, during fed state, fatty acids are synthesized because during fed state there are excess molecules available, so they are converted to or used for the synthesis of fatty acids. Now, what are some of the basic differences between fatty acid oxidation and synthesis? Now, there are a few differences. One of them is the location of the two processes. They are actually compartmentalized into two sections, two locations. The beta oxidation takes place inside mitochondrial matrix, whereas fatty acid synthesis takes place in cytoplasm. Another one is the carrier protein involved. Beta oxidation, we use coenzyme A, which carries the substrates involved in beta oxidation, whereas it is the acyl carrier protein ACP, which carries the substrates involved in biosynthesis of fatty acids. Another one is the fatty acid synthase enzyme, which is involved in fatty acid synthesis. It's a single polypeptide having a number of domains which catalyzes different functions, which takes up different catalytic functions. So this is unlike the enzymes involved in fatty acid degradation, where they have got there are different enzymes involved for different catalytic functions. Now, another important difference is the usage of NAD plus and FAD in beta oxidation, where it gets converted to NADH and FADH2. Whereas in fatty acid synthesis, which is basically a reductive biosynthesis, it involves NADPH. So these are some of the differences which you can go through. And fatty acid synthesis primarily occurs from glucose. So glucose, as you know, it shall be converted to pyruvic acid, gets into mitochondrial matrix, where it gets converted to acetyl-CoA enzyme. So as also some of the amino acids like leucine, lysine, the ketogenic amino acids, which are converted to acetyl-CoA enzyme in cytoplasm, as well as some of the amino acids which will get converted to pyruvic acid, and then pyruvic acid gives rise to acetyl-CoA enzyme again inside mitochondrial matrix. So one of the tasks of fatty acid synthesis is to take the acetyl-CoA enzyme, which is present in high concentration inside mitochondrial matrix, to cytoplasm. So this is one of the tasks, and this is actually carried out through citrate transport. There is a citrate transport mechanism involved in mitochondrial membrane, as you can see here. Yeah, this can be, yes, like this. So citrate, if it is available in plenty inside mitochondrial matrix, so citrate will be available in excess, obviously, when at fed state, when carbohydrates are in excess, it will be converted to acetyl-CoA enzyme inside mitochondrial matrix, which gives rise to citrate by combining with the oxaloacetate. And if citrate is in excess, because citrate do not want to be channeled to TCA cycle, so citrate can be converted to acetyl-CoA enzyme after it reaches cytoplasm. So obviously here you have to note that you don't need to consume excess of fat for conversion or storages of lipids. Even if you have excess of carbohydrates, they can be converted later to cholesterol as well as fatty acids and to other lipids also. So this is one of the aspects you have to note here. Now, citrate molecule gets into cytosol through citrate transporter mechanism, where it gets converted back to acetyl-CoA and oxaloacetate by a particular enzyme ATP-dependent citrate layers. Where acetyl-CoA now reaches cytosol, and it can be used for the synthesis of fatty acids as well as for the synthesis of cholesterol also. In cholesterol biosynthesis also, we have discussed this point in one of the earlier lectures. And what is remaining is oxaloacetate. Now, oxaloacetate cannot cross the membrane of mitochondria. It cannot go back to the mitochondrial matrix. So, it gets converted to malate at the expense of NADPH. Malate can cross this membrane of mitochondria to reach mitochondrial matrix and get converted to oxaloacetate. At the same time, malate can be converted to pyruvate, which uses the pyruvate transporter mechanism to reach mitochondrial matrix and get converted to oxaloacetate. Now, by the way, when malate is converted to pyruvate, NADPH or the reducing potential is produced, and this can be used for the reductive biosynthesis of fatty acids because it involves a number of reduction steps. And same as in the case of cholesterol biosynthesis also, NADPH can be used. And after all, cholesterol biosynthesis and fatty acid biosynthesis uses the same raw material as acetyl-CoA, which is actually derived from citrate here. Now, the product of fatty acid biosynthesis is the saturated straight chain 16 carbon palmitic acid, which is first synthesized, and all other fatty acids are actually produced by the modification of palmitic acid. Now, this is because fatty acid synthase, the enzyme involved in fatty acid synthesis, adds 2 carbon units sequentially, and it stops functioning when the chain length is 16 carbon. And the fatty acid biosynthesis occurs in cytosol of liver, kidney, brain, lung, mammary glands, adipose tissue. And the first step is involved is the committed rate limiting step. Now we actually move on to the process of beta oxidation, and the first step, the first committed step is shown here. This is conversion of acetyl-CoA, the 2 carbon acetyl group attached to CoA, conversion of acetyl-CoA to malonyl-CoA, the 3 carbon malonyl group. And this is undertaken by the enzyme acetyl-CoA carboxylase, which requires 3 things. Here, since this is a carboxylation step, carbon dioxide is provided. Energy is provided in the form of ATP, as also the coenzyme biotin is also needed for this particular enzyme. And studies reveal that this enzyme, it is active in the polymerized form. It can be monomers and it can be polymerized also. So the activation, that is polymerization, is favored by citrate, whereas polymerization is not favored. The equilibrium is shifted towards monomer formation by palmityl-CoA. Palmitic acid obviously is the end product of this whole process. Now further, the hormones like glucagon, epinephrine and norepinephrine, they favor or trigger monomer formation with phosphorylation through cyclic AMP mediated process of phosphorylation. Whereas insulin dephosphorylates the enzyme and tend to support polymerization of the chain, so that polymerization of the enzyme, so that the process, the beta oxidation, is favored by insulin. Now onwards, the malonyl-CoA is taken and acetyl-CoA is also used, and both they are actually linked to a particular protein called acyl carrier protein, or ACP. And the rest of the processes are actually catalyzed by an enzyme, fatty acid synthase, or simply FAS. And the fatty acid synthase is a unique enzyme in that it's a single polypeptide having 3 domains, and each domain having different catalytic functions in it. So this is a unique, unique enzyme having these functions. And let us see. So this is the structural representation of fatty acid synthase. It's actually a homodimer having 2 units shown here, but they are independent. And this single polypeptide, it contains actually 7 catalytic functions and 3 in 3 domains. Now this is the first domain having substrate entry and condensation units in it, which with the acetyl transferase, malonyl transferase, and keto-acyl synthase, the condensing enzyme in it. And domain 2 is the reduction unit, where acyl carrier protein is there, ACP is there. We will discuss more about ACP in the next slide. And there are beta-ketoacyl reductase enzyme, dehydratase enzyme, as well as enoyl reductase enzyme in this domain 2nd. And domain 3 is the palmitate release unit. It has thioesterase enzyme in it. Now, when coming back to ACP, the acyl carrier protein, the acyl carrier protein which is attached to this fatty acid synthase complex, it is having a phosphopentethene group attached to it like this, and the phosphopentethene group is having a sulfhydryl SH group also exposed to it here like this, and it is to this SH sulfhydryl group that acetyl-CoA comes and forms thioester bonds. As also malonyl-CoA also is accepted by this group, SH sulfhydryl group, so as to form malonyl ACP. So when we say that acetyl-CoA combines with ACP to form acetyl ACP, or malonyl-CoA combines with the ACP to form malonyl ACP, it is actually the ACP of this portion is being used by acetyl-CoA or malonyl-CoA to become acetyl ACP and malonyl ACP. And this is what I meant when I said the acetyl-CoA linking to ACP to give acetyl ACP, and malonyl-CoA linking to ACP to give malonyl ACP. So these enzymes, acetyl transacylase and malonyl transacylase, are present on the FAS or fatty acid synthase complex, so as to give acetyl ACP and malonyl ACP. Now the next step is the acetyl ACP and malonyl ACP are condensed to give acetoacetyl ACP with release of ACP. So this is a condensation process. Acetyl ACP combines with malonyl ACP to give acetoacetyl ACP with the release of ACP molecule. The enzyme involved is acyl malonyl ACP condensing enzyme, and this condensation reaction is actually driven by the release of this carbon dioxide by breaking the terminal this carbon of a malonyl group of malonyl ACP, which actually enhances the free energy of this reactants to a higher level than the free energy of the products of the reaction. So that's how the reaction is driven forward. And also note that obviously carbon dioxide expelled here is the carbon dioxide that is involved in the formation of malonyl CoA at the first committed step from acetyl-CoA. So as you can remember, acetyl-CoA combines with carbon dioxide to give malonyl CoA. So the same carbon dioxide is released here. So we can see that all the carbons of the fatty acids which is going to be produced come actually from acetyl-CoA only. Now when we say that acetyl-CoA is linked to ACP to form acetyl ACP, as well as malonyl-CoA is linked to ACP to form malonyl ACP, and further acetyl ACP and malonyl ACP are condensed to form acetoacetyl ACP. What actually happens here is that this FAS, the fatty acid synthase enzyme, is having, as I said earlier, the ACP unit with the phosphopentethene group having SH group is here. So phosphopentethene is not shown here, the SH group is shown here, as well as the FAS enzyme is having a cysteine also here, cysteine an amino acid, a sulfur containing amino acid, having a SH group, sulfhydryl group here. So what happens actually during condensation is that, now first of all, the acetyl unit comes and attaches, so this becomes acetyl ACP, and this acetyl ACP, the 2-carbon unit actually is temporarily shifted to cysteine sulfhydryl group. So there again a thioester linkage is formed, just as in the case of ACP, SH forms a thioester linkage with the 2-carbon acetyl unit. Cysteine SH also forms a thioester linkage temporarily with the acetyl unit. Now the malonyl unit comes, the 3-carbon unit comes and attaches to the ACP, so this becomes malonyl ACP. Now onwards, what happens is that the decarboxylation of malonyl ACP take place. That is decarboxylation, which is there are 2 carbons remain and 1 carbon is lost as carbon dioxide. And further, now the 2 carbon acetal unit here, so that is shifted to this position so as to complete the condensation and to give the final product acetoacetyl ACP like this. So this is the condensed acetoacetyl ACP, and now onwards we will continue with the process reduction, hydration and further reduction. So this is the first reduction step. This is acetoacetyl ACP gets converted to 3-hydroxybutyryl ACP, and the enzyme involved is beta-ketoacyl ACP reductase. And the reduction involves or use up this NADPH molecule. So this is actually the reduction of what is helped with the reduction potential, namely NADPH. And reduction actually occurs on this carbon, this 3rd carbon here. So this becomes hydroxybutyryl ACP. Now the next step is dehydration step, where hydroxybutyryl ACP gets converted to crotonyl ACP with the help of the enzyme 3-hydroxyacyl ACP dehydratase. Here again on the 3rd carbon here. So this actually a double bond is induced here with the dehydration to give crotonyl ACP. And the loss of our water molecules, obviously, since this is a dehydration step. And the final step is the second reduction. The final step in this first cycle is reduction of crotonyl ACP to butyryl ACP. Here 4 carbon compound is formed, and this is at the expense of NADPH and in the presence of the enzyme enoyl ACP reductase. And this reduction is important here. The removal of this unsaturated bond, that is double bond, is important here because the final product formed is an unsaturated palmitic acid. Now butyryl ACP, to butyryl ACP sequentially 2 carbon units are added. Obviously it is from malonyl coenzyme itself, but with the decarboxylation, and the site is the ACP site itself on the fatty acid synthase. So 2 carbons are added sequentially 1 by 1 till the chain length becomes 16, or 16 carbons are added totally. And fatty acid synthase actually stops functioning when totally 16 carbons are reached, which means palmitic acid will be released, and it's this enzyme thioesterase which is present on the 3rd domain of fatty acid synthase, that this enzyme is present which releases palmitic acid from multi-enzyme complex. And the sum total of reactions is that first one is one acetyl consummate, the initial one, 7 cycles of malonyl consummate totally, 14 NADPH, the reduction potential gives palmitic acid, and 7 carbon dioxide is released altogether, and 14 NADPH, 8 consummate, and dehydration 7 cycles, which means 7 H2O. And further 15 ATP are also used up, 8 during the process of citrate lyase catalysis. That is, citrate is converted to acetyl consummate and oxalate state in the presence of ATP dependent citrate lyase. So which uses 8 ATP for 8 acetyl consummate molecules, as also 7 ATP are used in 7 cycles of production of malonyl consummate. So this is the committed step, that is carboxylation of acetyl consummate to give malonyl consummate, during which ATP is utilized. And this is obviously we already discussed. And what is the regulation? Regarding regulation of fatty acid synthesis, acetyl consummate carboxylase is a rate limiting enzyme, as we have already seen. It's a monomer, but it can be polymerized, and the polymerized state is the active state. So citrate is a positive regulator for that. It shifts the equilibrium to the polymeric form, as well as palmitoyl coenzyme, which is actually the end product of the whole process. It's a negative regulator. It's a feedback inhibition sort of. It shifts the polymeric form to the monomeric form, or the inactive form. And high energy, that is high ATP ratio, ATP to AMP ratio, activates acetyl consummate carboxylase again. and that is by dephosphorylation obviously. And this are hormonal control. Regarding hormonal control, epinephrine, glucagon and norepinephrine phosphorylates the enzyme, which means that it shifts the equilibrium to the monomeric form. So it inactivates carboxylase. The phosphorylation is actually induced by AMP activated protein kinase. And insulin, whereas insulin is an anabolic hormone as we know, it dephosphorylates acetyl-CoA carboxylase. That way it shifts the equilibrium to the polymeric form and activates carboxylase enzyme. So this is insulin. It's an anabolic hormone. It favors fatty acid synthesis. And to add to hormonal regulation, insulin enhances fatty acid biosynthesis by basically enhancing the entry of glucose into cells. As also it activates pyruvate dehydrogenase, all of which will enhance the availability of acetyl-CoA. It actually inactivates lipases also, thereby reducing lipid breakdown. And insulin basically is released during fed state when ATP is high also. Whereas glucagon and epinephrine, they are catabolic hormones and they are secreted when ATP is low or energy charge is low. This table gives the entire fatty acid synthesis, that is a linen spiral after the discoverer Fedor Linen. It gives all the enzymes involved in a single cycle. Now, except for the first enzyme, this one, all other enzymes are part of FAS or fatty acid synthase. You can just pause this and go through. Thank you.